My dear student, colleagues, and all the viewers who are watching this program live from Facebook page and YouTube channel, I'd like to welcome you all to our International Physics webinar. Good evening to all here in Bangladesh, and a very good morning to all those who are watching this program live from USA. So hope you are well and safe from Corona pandemic. So we are trying to adjust with this new normal situation. Today is a very important day for our department. Today we are going to celebrate our 125th International Physics webinar. All we know that we are staying in a corona pandemic situation, so we cannot continue our normal academic program inside the campus. So we have to start our online program. I think you have already come to know that the Department of Physics, Pabna University of Science and Technology has started its online program, including International Physics webinar. We have successfully completed our 124th. So today is our 125th, and today I'd like to welcome you all to a joint session between the Department of Physics, Pabna University of Science and Technology, and the Department of Physics and Chemistry, MIT, USA, in physics. And we have with us here today Dr. Arup Chakraborty, sir, Robert T. Haslam, professor and chemical engineering and professor of physics and chemistry at MIT, USA. And he has already connected with us. So I'd like to welcome our speaker, sir. Good morning to your part and good evening to here. So welcome to our International Physics webinar, sir. Thanks for accepting our invitation.
Okay, should I start?
Sir, no, sir. I need to introduce you. So please give me a few minutes. So for those who are new, I'd like to introduce that we have divided our webinar into 3 parts. So first of all, I'd like to introduce our speaker with all of you, and then our speaker will deliver his piece with all of you. And at the end, we have a discussion time. In that time, anybody can join with us, and you can also ask question by commenting. I think you have already come to know the title of this today's International Physics webinar. Title is Understanding our Harnessing and the Immune System for Better Vaccine, a crossroad of physics and biology. And our speaker is Dr. Arup Chakraborty, sir, Robert T. Haslam, professor of chemical engineering and professor of physics and chemistry at MIT, USA. Dr. Arup Chakraborty is a Robert T. Haslam professor of chemical engineering, physics, chemistry, and biological engineering at MIT. He is the founding director of MIT's Institute of Medical Engineering and Science. He is also founding steering committee member of the Reagan Institute of MIT, MGH, and Harvard, and an associate member of the board of Institute of MIT and Harvard. After obtaining his PhD in chemical engineering and postdoctoral study, he joined the faculty of University of California at Berkeley in December 1988. He rose through the rank, the ultimate serve as the Warren and Catherine Slinger, Distinguished Professor and Chair of the Chemical Engineering, Professor of Chemistry, and Professor of Biophysics at Berkeley. He was also Head of Theoretical and Computational Biology at Lawrence Berkeley National Laboratory. In September 2005, Chakraborty moved to MIT. His entire career has been focused on research at the intersection of discipline, has received 4 teaching awards at Berkeley and MIT. After an entire career in engineering and polymers and catalyst since 2000, Chakraborty's work has focused on bringing together immunology and physical and engineering sciences. So more specifically, the intersection of statistical mechanics and immunology. His interest span T-cell signaling, T-cell development and repertoire, and the mechanistic understanding of HIV evolution, antibody evolution, and vaccine design. Chakraborty's work at the intersection of discipline has been recognized by numerous honors, including the NIH Director's Pioneer Awards and EO Lawrence Medal for Life Sciences from the US DOE, the Alan P. Colburn and the Professional Progress Award from the Alchemist, a Dreyfus Teacher Scholar Award, and the National Young Investigator Award. Chakraborty was elected a member of the National Academy of Sciences and the National Academy of Engineering for completely different bodies of work. He's also member of the National Academy of Medicine, making him one of the small member of the individual who are member of all 3 branches of US National Academies. He's also a fellow of American Academy of Arts and Sciences and American Association for Advancement of Science, and served on the US Defense Science Board. Chakraborty has received 4 teaching awards at Berkeley and MIT. We can see his interest: statistical mechanics, molecular and cellular immunology, immune response to HIV and influenza, vaccine development, gene regulation, and in mammals, teaching, and his awards and honors. So his publication: over 2015 publication, and over 430 invited lectures, 1 co-authored books, and 1 entitled books. Thanks all for all your patience. Now it's time to go to your speaker. Sir, thanks again for joining with us. It's your time, so you can start your session, sir.
Okay. Can you see it now?
See.
Can you see my screen?
Yes, sir, we can see.
Okay. So thank you very much for the very generous introduction, and I'm pleased to be here to share some of our work with you.
Yeah, it's my pleasure, sir. You can start.
As was mentioned, the vast bulk of my work is focused on trying to understand how the immune system works and then trying to harness that knowledge towards practical ends like better vaccines. So by way of introduction, let me say that we have an adaptive immune system, which is remarkable in that it enables us to mount pathogen-specific responses against a diverse and evolving world of microbes. Think about how amazing that is. That is, you can mount a response that is specific to the SARS-CoV-2 virus that causes COVID-19, which evolved after you were born. To roughly see how that works, consider infection with a virus. So a virus infects a host cell and hijacks its transcriptional machinery, enabling the synthesis of viral proteins, which can then make many different virus particles that can then go on to infect other cells. Important players in adaptive immunity are B cells. We have about 100 billion B cells, and most B cells have a receptor on their surface that is distinct from the receptor on the surface of another B cell. If the receptor on this particular B cell can bind sufficiently strongly to the spike proteins of this virus, biochemical reactions cause it to get so-called activated. It starts to proliferate, make many copies of itself, and a Darwinian evolutionary process ensues in a very short period of time, which I will describe later, which ultimately leads to secretion of soluble and even higher affinity forms of this receptor, and that is what we call antibodies. These antibodies can then bind specifically and strongly to the spikes of this particular virus and then dispose of it in a number of different ways. But antibodies principally do battle with virus particles that are in your blood or extracellular spaces. Now, some of these viral proteins are chopped up into short bits called peptides, about 9 to 10 amino acids long for our purposes today, and they are displayed on the surface of these infected cells in complex with human proteins called MHC proteins. Now, we each have 6 to 12 types of these MHC proteins, but there are thousands of variants in the human population. Different MHCs tend to bind different types of peptides. T cells are also important players in adaptive immunity. Again, we have about 100 billion T cells, and most T cells have a distinct receptor on their surface, and if the receptor on this particular T cell can bind strongly to this particular viral peptide, then biochemical reactions activate it, it starts to proliferate, and then it can coordinate an immune response in a number of different ways. For example, certain kinds of T cells called CTLs are such that if an activated CTL sees another cell displaying the same peptide that originally activated it, it will secrete products that will kill this infected cell and the virus particles that they harbor. Now, after you have cleared an infection, most of the B cells and T cells that proliferated in response to this particular infection die, but a few remain as so-called memory cells, which can mount robust and rapid responses on reinfection with the same pathogen. This pathogen-specific immunological memory is the basis for vaccination. With a vaccine, you try to induce memory B cells, T cells, and antibodies that are specific for the bug against which you wish to protect the host. Now, vaccination has saved more lives than any other medical procedure, and its importance has been made vivid in the times we live in, where the COVID-19 pandemic continues to rage and hope is offered by the new vaccines. Now, SARS-CoV-2, this virus, has spikes on its surface, depicted here in red, And these spikes bind to human receptors or proteins on the surface of our own cells. In particular, this virus binds to the receptor called ACE2, which helps regulate blood pressure and is abundantly present on cells in the lung, intestine, heart, kidney, etc. Once the spike binds to this ACE2, then SARS-CoV-2 has evolved such that one of our own proteins, which I have depicted here as a scissor, can cut the spike into two. And that's like releasing a spring, which allows the virus to enter our cells, deliver its genomic material, and begin to replicate. So if you can block the ability of the virus's spike to bind to ACE2, you will help prevent infection. And indeed, all vaccines efforts today are focused on eliciting antibodies that can bind to this region of the spike protein of SARS-CoV-2 to prevent infection. The Moderna and Pfizer vaccines use a certain kind of technology that has never been deployed before, and the Johnson & Johnson and AstraZeneca vaccines use another kind of technology that had never been deployed before. Now, SARS-CoV, all these vaccines are enormously effective, and part of the reason is that SARS-CoV-2 is a virus family that does not mutate that fast. Although you are hearing a lot about the mutations, they are not as fast. Imagine if in the future a pandemic was caused by a virus that could be spread by casual contact, like COVID-19, but which also mutated very, very rapidly. We don't have to imagine this. Indeed, the HIV pandemic is caused by such a virus. It is not spread by casual contact, but it is continues to wreak havoc in many parts of the world. In order to eliminate scourges like this from the planet, we must design vaccines based on a rational mechanistic understanding of the underlying virology and immunology. Now, a major barrier to the quest for a vaccine against HIV is the high mutability of the virus. So in these phylogenetic trees, the number of N's that you see corresponds to the diversity of the virus. And you can see in this diagram to scale that the diversity of the flu virus in the entire world in a particular year is comparable to the diversity of HIV strains in a single infected individual, and completely dwarfed by the diversity of strains in just one region in Africa. This high mutability presents a challenge for vaccination because HIV can mutate to evade vaccine-induced antibodies and T cells that lie ready and waiting for certain specific strains. Today, I'm going to briefly describe to you some of our work aimed to address these challenges, both for the antibody and T cell arms, beginning with our work on T cells. Because of the high mutability of the virus, there has long been interest in focusing a vaccine-induced T cell response to target single residues on HIV proteins that appear relatively conserved if you line up sequences of the same HIV protein derived from virus samples extracted from diverse patients. The idea, of course, is that if a T cell attacks such a residue that appears relatively conserved, that is, the amino acids don't change much as you go from one patient to another, then HIV would have to evolve a mutation there to evade the T cell pressure, but that should likely make the virus unfit, because why else do you see that particular residue's amino acids to be relatively conserved? But this strategy can be blunted because of the high mutability and replication rate of HIV. It can often evolve other mutations, so-called compensatory mutations, that partially restore the fitness cost incurred by making the first immune-evading mutation. So now you have a virus that's almost as fit as before, and it has evaded the T cell pressure. The point is that if you want to define the mutational vulnerabilities of a virus like HIV, you have to define these collective compensatory pathways that the virus uses to evade human immunity in order to not target them with a vaccine-induced T-cell response. And you also want to define the combinations of mutations that the virus cannot make and remain viable in order to target them with a vaccine-induced T-cell response. Or in short, you want to define the fitness landscape of the virus. To make sure we are on the same page about what I mean by a fitness landscape, let me first illustrate this on this two-dimensional sheet using a cartoon protein that is made up of only two residues. So in this cartoon diagram, each grid point on this axis represents a different amino acid that could be present in residue 1, and each grid point on this axis represents a different amino acid that could be present in residue 2. So for this cartoon protein, any point in this two-dimensional space represents a mutant viral strain, and information about its fitness, that is, its ability to replicate and propagate infection in vivo, is depicted on this third axis. So you can see how this information about fitness as a function of sequence will trace out hills and valleys, but of course in a much more higher dimensional space than what I have illustrated here. Now, knowledge of this fitness landscape can be very useful for vaccine design, because with a vaccine-induced immune response, you want to push the virus off of one of these fitness hills and force it to make immune-evading mutations that correspond to one of these fitness valleys. And you also want to block these mountain passes that correspond to the compensatory mutational pathways that the virus uses to go from one hill to another adjacent one when this one is under attack. So armed with this fitness landscape, you can design the immunogen, or the active component of the vaccine, in a way that does not contain the whole proteins of the virus as is typical, but only parts of the proteome of the virus. So you minimize regions that are rife with these kinds of compensatory pathways. So then if it's not in your vaccine, your immune response will not target such regions. You maximize regions where multiple mutations are especially bad. So if that's in your vaccine, then your immune response is likely to target it. And remember what I said about many diverse MHCs in different people. You maximize regions that can be presented by people with different genotypes of MHCs. It is this very practical goal that led us to try and translate the data on thousands of sequences of HIV proteins that have been collected from diverse patients over the years into knowledge of these fitness landscapes. This work has been going on for some years, and suffice it to say that the people in the top row were former students and postdocs who are now faculty members at various universities, and also others who are even more gainfully employed. And I will mention these people in context. So the way we try to obtain this fitness landscape of the virus is by starting with a more modest goal, that is trying to find the prevalence landscape, that is the probability of seeing a particular sequence represented as a vector Z in circulation. The sequence is represented as a vector because it's just a list of amino acids of the different residues of the protein. Now, this information of the probability of observing a sequence in circulation is contained in the sequence data. The sequence data, of course, can also be used to infer the probability of seeing single mutations at every residue, double mutations at every pair of residues, triple mutations at every triplet of residues, and so on. Any mathematical model that can recapitulate these mutational correlation functions is a legitimate model for P of C. So to do this, we adapted and extended approaches that had been developed in other contexts and asked, what is the least biased model for P of Z that by construction fits the observed 1 and 2 site mutational probabilities? And then exploiting the connection between statistical mechanics and information theory, least biased is interpreted as the maximum entropy model. So let me illustrate this. If Zk is a vector characterizing the kth sequence, then the probability distribution that we seek is P of zk. Its entropy is given thusly. We want to maximize this entropy subject to the constraint that the probability distribution is normalized, and the predicted 1-point mutational probability is the observed one, and the same story for the predicted 2-point mutational probability. These constraints are imposed as Lagrange multipliers, and 2 lines of algebra will show you that the formula obtained for P of z then is given by this familiar form from Mr. Boltzmann, with the Hamiltonian given like so, where these fields reflect the difficulty of making mutations at a residue I, and these couplings reflect the effects of coupled mutations. So if Jij is positive, mutations at sites I and j are compensatory. If Jij is negative, then mutations simultaneously at sites I and j will be even worse than independent. But at the end of the day, the model we have obtained is a model for prevalence, circulation prevalence. Now, you could argue that the more prevalent strains are also likely to be more fit. Otherwise, why else are they there more? But this argument could be very complicated, because the sequences we have used to infer this P of C were not sequences that were circulating, or that were grown in Petri dishes. They were extracted from humans, where there was a battle between the immune response of this person and the virus. So the virus may have evolved a mutation to escape the immune response of a particular person, and this mutation made the virus intrinsically less fit, but because it avoided the immune response in this person, it was effectively the fittest virus. We seek the intrinsic fitness landscape of the virus, because then we want to learn how to push and pull on it with a vaccine-induced immune response, so that the virus is cornered between either being killed by the immune response or evolving suicidal mutations. So with these 2 gentlemen, using a variety of approaches from the physical sciences, we explored this connection between the fitness and prevalence landscapes of HIV virus. And the bottom line of all these studies was that the fitness and prevalence landscapes are not the same, but for HIV, the rank order of the prevalence and the rank order of the fitness are statistically similar. And that is all we seek, that we need to know this strain is more fit than this one, and this one, and this one. Let me briefly show you one approach that we took to arrive at this conclusion. This is Eigen's famous equation for evolution of a quasi-species of a virus, where X alpha is the frequency with which you observe strain alpha, and this is its time evolution. So it depends on the fitness of the strain, how quickly it replicates, but the fitness is compared to the average fitness of the prevailing other strains, because there's competition between them. And this is just mutations away from strain alpha to beta, and this is mutations in from strain beta to alpha. Several years ago, Lighthouse showed that this non-equilibrium dynamical equation is isomorphic with the equilibrium statistical physics of a 2-dimensional Ising model. In this Ising model, any row here is a viral strain. These arrows represent what the amino acid is here. And the next strain, the next row, is the progeny of this strain, and so on. Now, any given configuration of this 2-dimensional Ising model is therefore an evolutionary trajectory. So this is like a path integral description, if you like. And for our case, the Hamiltonian that describes the weight of a particular evolutionary trajectory is shown like so. It has in-row couplings that reflect the intrinsic fitness of the virus, and modulated by immune response, which acts like an external field to promote mutations. And across rows, there is a nearest neighbor ferromagnetic coupling, which simply says, physically, that if I have an arginine here, my child is likely to also have an arginine here because the mutation rate is less than half. Now, once you have a Hamiltonian, you can do many things with it, exploiting the machinery of statistical physics. For example, here is the real Hamiltonian for this Ising magnet. The Hamiltonian we infer for the fitness is of this quadratic form. And one way we can do this is just use Feynman bounds to ask, what are the best formulas that you can obtain that describe the dependence of the inferred couplings and fields as a function of the intrinsic fitness, the immune response, and the mutability of the virus? Let me not take you through these formulas, but rather just say what this analysis revealed in terms of biological reasons as to why the relationship between fitness and prevalence is relatively simple for HIV. As I mentioned to you, HIV is a chronic infection. There is a great diversity of MHC genes, so different peptides are presented by different people, and that's what T cells attack. And so if you go look at the clinical data, you will see that no particular region of HIV proteins is targeted by a significant fraction of people because of the great diversity of genotypes amongst us. Also, if you look at the clinical data, you will see that if I force the virus to make a mutation because of my T cell response, and that mutation incurs a fitness cost, and then I infect another person who targets a different region, then because HIV is a chronic infection, over time, that fitness costing mutation will revert back to its original form. So coming back to what it was. And very importantly, unlike influenza, the HIV population in the world has never been subjected to a few classes of effective, natural, or vaccine-induced memory immune responses. So the HIV population is not evolving in narrowly directed ways to avoid past herd immunity in the population. And you can see this vividly if you look at the phylogenetic trees of influenza versus HIV. The influenza tree looks like a flowing river with little branches coming out of it, whereas as I showed you, the HIV phylogenetic tree is like a dendromer. So for these reasons, while human immunity forces the virus to sample sequence space, at the end of the day, it's sort of like a temperature that shakes it and does not change the rank order of prevalence and fitness. But these arguments and these calculations and the machine learning inference that we did are all approximate. And so they could be completely wrong about such a complex problem. And the only way to test their veracity is to subject predictions coming out of this theory against in vitro and clinical data. And let me show you snapshots of that, beginning with in vitro experiments carried out in the lab. So we can make predictions of the fitness of different mutant strains, and our collaborators can then make the same strains by site-directed mutagenesis and test our predictions. Now, the metric of fitness in our model is E, the Hamiltonian corresponding to a mutant strain. And the model suggests that the value of E for a mutant minus that for a reference strain should correlate negatively with the fitness of that strain divided by the fitness of a reference strain plotted in these logarithmic coordinates. So I'm first going to show you some experimental data that were obtained to test our predictions by Tumbi Udungu in South Africa and Bruce Walker here in Mass General Hospital. What they did, and in particular, we'll look at predictions where we made predictions for mutations introduced into a large polyprotein of HIV called GAG. We made predictions for several strains, 39 of these, and here is the data. The data are the points, and this is the prediction. And only 6 of the data points are not shown here because we predicted that they would be extremely unfit, and they indeed did not grow out at all. And the experiments that were done as follows: they take the virus, put it into a Petri dish with human cells, and they watch its ability to infect these human cells and grow out. And these ones that we predicted to be extremely unfit did not grow out at all. And so the replic fitness would be 0, and it would be difficult to plot that on a logarithmic scale. Nonetheless, including those strains, clearly we are not perfect, as you can see, but with high statistical significance, our predictions capture about 80% of the data. Here's another comparison. This is with another large protein of HIV envelope, and here are over 100 data points. And again, although there is scatter around our prediction, we don't do too badly. Comparisons with other HIV proteins in this way are also encouraging, but rather than show you that, let me show you one snapshot of comparison with actual data in humans, which we did with Andrew McMichael's lab. So Andrew had historical blood samples from HIV patients that had never been on drug treatment before. And he had these blood samples as a function of time in these patients. So he could sequence the viral strains in each patient as a function of time. And he also knew where the T-cell response of each of these patients was targeting the virus. In these patients, the virus was always able to make a mutation to evade the T-cell pressure. And so Andrew asked, based on your fitness landscape, can you predict that of all the places where mutations could have emerged to evade the T-cell pressure, where did they actually arise in these patients? And can you predict the relative times at which these escapes occurred in these individual patients? For over 100 years, since Wright and Fisher, people have been doing stochastic evolutionary dynamics, but to our knowledge, with the fitness landscape being cartoon ones. To answer these questions, we did very similar calculations, but now with the fitness landscape of a real human pathogen under immune pressure. That is also real. So the input to this calculation is the initial swarm of sequences of viruses and the locations that the T cells are targeting in each patient. So how do we do with predicting where the mutations emerge? Well, we don't do too badly. We get it right about 86% of the time. Rather than showing you this data in detail, let me illustrate how our results show or highlight the importance of these collective compensatory pathways that the virus uses to evade human immunity, which also highlights the importance of the sequence background in which the same mutation has to emerge. To illustrate this, it is best to compare two patients that target precisely the same region of the virus with their T-cell response. So these are two patients who targeted exactly the same region of the virus. They escaped by precisely the same mutation, but in this patient, this escape mutation arose 40 times more slowly. This circle represents the list of the protein from which this peptide is derived, and the marked residues are those where mutations existed in the virus that infected this patient. Blue lines indicate that we predict that the pre-existing mutation has a compensatory effect, so it makes it easier for the escape mutation to emerge. Red lines indicate the opposite, that is, the pre-existing mutation makes it even more challenging for this escape mutation to emerge. The thickness of the lines is the value of Jij, or the effect of the strength that we predict. As you can see vividly in this case, this patient was infected with a virus that contained many more pre-existing mutations that were strongly negatively coupled with this escape mutation. So it made the escape mutant more unfit, and therefore it took much longer to grow out and take over the population compared to this guy. How do we do with predicting the relative times at which escape occurs? In these evolutionary dynamics, we can predict time only in generations of evolution, not in actual time. But here it is. This is our prediction of the escape time in different patients. This is the escape time measured in days for these patients. And here is the comparison. And you can see there is plenty of scatter, But nonetheless, with high statistical probability, we do not do too badly. And please remember, these are not contrived experiments, but these are the virus evolving naturally in these people. So we also have looked at other ways to compare against patient data. For example, there are a few people with special genomes called elite controllers who can control the virus to low levels without any treatment. And we find that, according to us, the regions of the HIV proteins that they target with their T cells are multidimensionally conserved. By that, I mean that it's very difficult to evolve mutations there, regardless of the sequence background. So based on these sorts of comparisons and others that I have not shown you, we decided to design this immunogen made up of only parts of the proteome, which are regions where compensatory mutations are not very likely, and antagonistic mutations are there potentially, and it's also very difficult to mutate there regardless of the collective effects. Now, the trouble is that at the end of the day here, we get short peptides. And so how to deliver them in an immunogenic fashion, that is the one that will raise lots of immune responses, is a challenge. And one way to do that is to use adenovirus vectors. So these are viruses that have been engineered to not replicate. And they are cold viruses, cause common colds and so forth. So they cannot cause disease, but you can insert your piece of the genome corresponding to HIV proteins that we have designed into this. And then this will infect human cells and raise an immune response against the proteins of HIV that will be made. By the way, this is the technology that is used in the COVID-19 vaccines by Johnson & Johnson and AstraZeneca. So the problem is that the insert containing the HIV protein has to lie in a certain range for stability. So Darius Murakowski and I designed the so-called greedy algorithm to include as much of the good bits, that is the parts that are mutationally vulnerable, without creating too many unnatural junctions. Because remember, we get short bits, we have to join them. The reason we don't want too many unnatural junctions, because an unnatural junction is one that is not present in HIV proteins. So if we get an immune response to that, it's worthless. So this is what we did. This designed immunogen of 551 amino acids was inserted into this non-replicating viral vector AD26 by Dan Baruch at Harvard Medical School, who is the creator of the Johnson & Johnson vaccine for COVID. And this is exactly the platform that's used there. And then he immunized monkeys with this. And here is the monkeys. This is a small study, only 4 monkeys. This is the monkeys that were immunized with our vaccine. And this is the 2 monkeys that were immunized with the full protein, for which Dan has hundreds of monkeys' data. This is the size of the immune response that you get. And you can see that our construct is just as immunogenic as, raises just as much of the immune response as the full proteins, but ours contains only the vulnerable bits. So this is after 1 shot of the vaccine. Look at the scale here, 700. And this is after the boost, becomes 7,000. And again, we are just as immunogenic as before. So we are now beginning to think about how one might try to use this vaccine in a therapeutic sense for HIV, but that is a very challenging path forward. But at least we are happy that we have come from statistical physics to monkeys in this project so far. In the remaining few minutes, I want to very briefly tell you about some work we are doing on the antibody side of this problem. And I will be very brief here because of time is passing by. This is the viral spike of HIV. I told you that it mutates very rapidly. However, it contains some regions, such as the one shown in yellow here, that is relatively conserved because it has to bind to human receptors in order to propagate infection. but it is surrounded by these highly variable regions shown in red and protected by these sugars shown in blue. Now, immunologists have been able to isolate antibodies from infected people that at least in vitro can neutralize diverse strains of HIV. These so-called broadly neutralizing antibodies, or BNAbs, bind to these relatively conserved regions. The trouble is that these BNAbs emerge in these patients several years after infection, and that too in low numbers, so they're not protective for this particular person. Nonetheless, they provide proof of concept that the human immune system can evolve such BNAbs and raise the tantalizing possibility that properly designed vaccination protocols could induce such BNAbs in diverse individuals effectively. Now, antibodies are produced upon natural infection or vaccination by a Darwinian evolutionary process called affinity maturation. I told you that early in the talk that if a B cell has a receptor that can bind sufficiently strongly to the spike of a particular virus, it starts to replicate. But it can also at that time seed structures in your lymph nodes called germinal centers. Here, as the B cell proliferate, mutations are introduced into the regions of their B receptors that bind to the antigen at a very high rate. These mutated B cells then interact with the virus that is presented on other cells in the lymph node, and the B cells that bind more strongly to the spike of this virus, they can internalize more antigen. So if the B cell binds to this antigen, there's a pulling force that acts and can eat the antigen. If the binding is not strong enough, this pulling force will not eat antigen that much. Then these B cells can chop up the proteins of the virus, present them as short peptides, and then B cells that have eaten antigen, they compete with each other to interact with another kind of T cell called a helper T cell. And productive interaction leads to a survival signal. Otherwise, the B cell dies. So you can see B cells with receptors, mutated receptors that bind more strongly to the virus are likely stochastically to eat more antigens, likely stochastically to present more antigen, and stochastically more likely to win this competition and survive. So now a few of these positively selected B cells are let out as memory cells and plasma cells, which then secrete this receptor, and that's what's an antibody, as I told you. But the vast majority of these are recycled for further rounds of mutation and selection. So this is a problem. To understand this problem is really a problem in non-equilibrium statistical physics and its intersection with immunology and learning theory, because now what you want to do is put as the antigen different kinds of vaccine antigens that will teach this evolutionary dynamical process to evolve generalists, that is, those that bind to diverse strains of BNAbs, and not specialists that bind to only one strain. You can see the induction of these broadly neutralizing antibodies will require vaccination with more than one strain. Because if you give just one strain of the spike, you will get a very strain-specific antibody due to this Darwinian process. What you want to do is immunize with multiple strains whose variable parts are very different from each other, but which share these conserved residues that you want this evolutionary process to learn how to target. So once you have to give many variants, you have many questions. What should be these variant antigens? How many should there be? How different should the variable regions be? Should I give all of the antigens as a cocktail, or should I give them sequentially, and so on, so on? The answers to these questions are drawn from a huge space of possibilities, and just randomly doing experiments with monkeys may not be enable us to find the golden nuggets that we seek. So you might say that knowledge of affinity maturation can guide answers to these questions. But the trouble is, until very recently, this process had been studied only in the presence of single antigens, so how it occurred when there were multiple variant antigens was poorly understood. That is why a few years ago, we started to build physics-based computational models to understand this basic problem in immunology, which might also help us guide vaccination strategies. This too has been going on for 5 or 6 years and has involved collaborations with many young people who have gone on to their own careers, involved collaboration with a number of biologists and physical scientists. So the competition model, of course, is very simple. All you have to do is that after activation, you get replication and mutation, affinity-dependent selection, a few are let out, and there are many rounds of mutation and selection. But there are 2 boundary conditions to this dynamical process. Actually 3. One of them is an absorbing boundary condition. All B cells die. Extinction is always possible in an evolutionary process. The other is that the number of B cells undergoing this process exceed a threshold. This is just a proxy that as B cells eat antigen, if they keep multiplying, they will soon eat up all the antigen, and that will bring it to a stop. And there is a third boundary condition, which is a maximum time, because antigen decays on its own. So you can model this non-equilibrium dynamical process with a number of different ways: stochastic agent-based computer simulations, Fokker-Planck or master equations, and whichever way we have done it, at the end of the day, what you get, I'm not telling you these because time is short now, what you get is the probability of obtaining antibodies of different breadth, or the number of BNAbs produced with different strategies. Again, just a summary now, without any details. If you give a cocktail of variant antigens that are separated by large mutational distances, what you see is most likely the B cell populations just die. Why? The physical reason is illustrated here in this cartoon. These are 3 variant antigens that share conserved red regions, but they are different variable regions as depicted by these different colors. Now imagine that in a particular round of mutation and selection, this B cell is positively selected by this antigen. Now in the early rounds of affinity maturation, the affinity of this receptor for the antigen is not very high. That is why it goes through this affinity maturation process. So the chance that its binding affinity for the shared conserved residues is high is also low. So in the next round of mutation and selection, if it has to be selected by this guy, the chance that it will bind strongly is not high, and therefore it is likely going to die, or apoptosis, as the biologists say. The point I'm making is that these variant antigens are present simultaneously are conflicting selection forces that can frustrate this affinity maturation and result in death, which is an absorbing boundary condition. We have found that sequential immunization with the same antigens allows the system to learn how to evolve BNAbs better. And this has been tested against experiments. So what do I mean by this? The first antigen you give results in the formation of memory B cells that have adapted to the best they can to a variant antigen, which is the selection force. Upon vaccinating with another variant antigen, this existing memory B cell population is thrown out of equilibrium or steady state. So now a non-equilibrium dynamics occurs. The population tries to adapt. And that's what I mean by sequential immunization. So the question that was raised then, in each of these vaccinations, how far off of steady state must we kick the existing population off so that adaptation to develop broadly neutralizing antibodies is optimal? Again, without details, because of time. The work of these 2, she's no longer a postdoc, she's a faculty member at University of Colorado, and Raman Ganti, who is a postdoc with me for the moment. Here is what we have found. If we have too low a mutational distance between the second and first antigens, the resulting non-equilibrium force that leads to the flux towards adaptation is small. The population rapidly multiplies, and you get lots of memory cells, but they don't adapt much to the new antigen. And so now when you come with the next one, forcing them to become BNAb, most of them just die. If you have too high a mutational distance between the second and first antigen, the non-equilibrium force driving towards adaptation is too large. Normally, you would expect that therefore the flux would be very high. But because of the absorbing boundary condition of the other side, it starts to go in the wrong direction, and the whole B cell population is extinguished. So there is an optimum mutational distance between the first and second antigens that leads to the right kind of B cell diversity we find, so that the third antigen, there are many paths to success. There is also an optimum for the subsequent variant antigens, and they must drive the system further off of equilibrium than the previous optimum. Anyway, I know that these are not very clear to you because I was very fast in the end, but I did want to give you a flavor of this effort on antibodies, because it's also work that you can do for flu, like we are doing. It may also be relevant if the coronavirus population begins to mutate in a directed way because of global vaccines and so forth. And more importantly, from a physics point of view, it is, in my opinion, a very interesting problem at the intersection of evolutionary biology, immunology, non-equilibrium statistical physics, and learning theory. So let me end by simply saying this. Affinity maturation, this Darwinian process that leads to antibody production, is a non-equilibrium dynamical process. And one way to think about it is that it has two very strong attractors, a strain-specific antibodies and a value of depth. And there's a narrow ridgeline to this broadly neutralizing antibodies. And what we are beginning to suggest from our work is that there is some optimal immunization strategies that can try to convert this narrow ridgeline into a broad stairway to heaven, if you like. So with that, let me stop. And I'm happy to answer a few questions in the remaining time. Thank you very much.
Thank you, sir, for your wonderful presentation and very informative. So I think students have learned a lot of things about vaccination, and it is very important for this COVID situation. So we have got a few questions in the inbox, and we have a guest with us. So I would like to add one of our guests. So Angon, can you hear us?
Yeah. Good morning, sir.
Okay, so if
good morning. Please call me Arup.
Okay, thank you, sir. Sir,
so thanks for joining with us. If you have a question, you can ask.
Yeah. Sir, can I ask, please?
Yeah, of course you can ask.
Yeah. Sir, my first question is, except COVID-19, which viruses have spike protein?
I didn't hear you. You said what about COVID?
Sir, except COVID-19, which viruses have spike proteins?
So most viruses have spike proteins. HIV has a spike protein, as I just described, and the antibodies target that. Influenza has a spike protein. So for example, I don't know if you can still see my screen, but if you can, this is the spike protein of influenza. So viruses have a spike protein for a purpose. They use the spike protein to bind to human protein so that they can enter cells. So viruses have spike proteins so that they can enter other cells.
And sir, my second question is, sir, does HIV virus also similarly binds with human cells like COVID virus?
Yes. So the HIV spike binds not to ACE2 like the SARS-CoV, but it binds actually to proteins on our immune cells. And it infects the immune cells, like T cells and other cells called dendritic cells. And as a consequence, it starts to kill our immune cells. And that is why, when you are infected with HIV, you become immunocompromised, because many of your immune cells are killed rather than lung cells and so on.
Thank you, sir, so much.
Thank you, Angon, for joining with us. So, sir, we have some question in inbox. The first question, sir, if somebody achieve herd immunity, is he or she need to take vaccine?
Well, first of all, somebody does not achieve herd immunity. A population achieves herd immunity. So, and the idea there is very simple. Suppose that The concept of herd immunity is very simple. And I think I'll tell you how I described it in my recent general audience book that I have written. So what basically is happening is that imagine that there is a virus that has infected me, and this virus is such that on average, during the time I will be infected, I will infect 3 people. Okay? So when nobody has immunity, I will infect 3 people. But now, if different people start to get vaccinated or infected, such that they have immunity now, so I will still interact with 3 people. But if they all have immunity, then I will infect no one, and so the propagation of the infection will stop. And roughly speaking, if the virus is such that I infect 3 people on average in an infected period, then the herd immunity will be achieved when 1 minus 1/3, so 2/3 of the population is infected, is immune. That is what we mean by herd immunity. So herd immunity is for the population, not an individual.
Okay, sir. So there is another question. So can competition or model predict the success rate of our vaccine?
Well, as you can see, we are trying to take the first steps towards designing such vaccines, but then as to how it will work in a broad population is a matter of debate. But what I'm trying to show you is that very physics-based models and computations can help you make virology and immunology a more physics-like discipline rather than empiricism alone.
Thank you, sir. This may be the last question. So as we are taking the vaccine, so whole world are taking, so how long the COVID will exist after taking this vaccination?
So first of all, it will depend upon how we deploy these vaccines and how quickly we deploy them. But remember that this will not be a story like smallpox.
Yeah.
And the reason is that there is a viral population in animals. So it can jump again and so forth. So.
Thank you, sir, for your wonderful presentation and nice discussion session, sir. So very informative and very actually interesting topic you have talked with us, and I think our students have learned, and those who presented today's lecture and all have learned a lot of things about vaccine and this system. So thanks again for accepting our invitation. So bye for today. Hopefully we'll arrange another webinar with you, sir. And you have given us time from your valuable time. We know you are too busy with your works. So thanks again, sir. We'd like to say thanks on the behalf of the Department of Physics, Pabna University of Science and Technology, sir. Bye for today. Good night.
Dhanyavad.
Dhanyavad, sir. Apnake. Namaskar.